very good. Okay, so our next presentation is Automated Negative Gene Ontology Based Functional Predictions for Proteins with UniGOPred. Oh, it's a mouthful. Tunca Dogan.
Thank you very much. Today I'm going to talk about negative functions of proteins. First, I should define what negative function is. It can be defined as all those functions that is not possessed by a specific gene or protein. And why it's not trivial to work with negative functions? Because first of all, function annotation of biomolecules in databases are incomplete. And when you come back to the database, annotation database 6 months later, when you check your favorite protein, then you see that additional Go term annotations, for example, has been added. And in this toy example, I want to mention that these nodes are corresponding to genes or proteins in our protein space, and according to a target Go term function, the red ones are representing the ones that do not possess that specific function, and the blue ones are the ones that is having that specific function. So these are the true labels of these proteins, but in any case, at any time, we don't have this information. We only have an information something like this. We only know the some of the proteins that has the function. We do not know anything about the negatives. So when you want to develop an automated protein function prediction method, the classifier usually does not know where to separate these two classes from each other. So it can be from here, from there. So there is no information, no clear information, which reduces the performance of the automated protein function prediction methods. But is there any way to have at least some of the negative samples? Then we could use this information in numerous ways. For example, it can be useful in planning systems biology research-focused wet lab experiments, because you have some hypothesis to begin with, and it can help forming that hypothesis. And second of all, it provides ideas about positive functions as well. Going back to our toy example, when we have a few negative samples at hand, then a simple binary classification method can separate them accurately to predict new proteins that have the function. And also, there's an interesting idea. It's possible to propagate negative annotations to the child terms, just similar to how we do the regular positive annotations, propagate them to up to the root, following the true path rule of directed acyclic graph of Go. So if we have, for example, a negative annotation for these two terms on the gene ontology graph, then we can safely propagate as a negative annotation the child term as well, and we can enrich our negative annotation data set. And one last thing would be, it can be helpful in loss of function studies, which I will mention in the results part. So do we have negative function annotations in biological databases? We have in UniProt, there are more than 23,000 not qualifier annotations, and these are used where associating a Go term with a gene product should certainly be avoided. And here is an example for a human protein. It has been annotated with positively annotated with a cellular component Go term, and also not annotated with another one, a sibling term. But their number is quite small, so we should enrich them. We can predict them. And here we tackle this problem by developing a new method called UniGOPred to produce negative functional predictions as well as the regular positive functional predictions. So I don't want to go into the methodology, just to say that our tool takes the protein sequence as the input and outputs the Go terms together with prediction scores that is ranging between 0 to 1. So here, in order to produce accurate negative annotations, the training data set generation is crucial. And for each Go term, we have positive and negative training data sets. Preparation of the positive training data set is trivial. We just take the proteins that is annotated with that Go term from UniProt SwissProt database. And for the negative training data set generation, we stay away from the trivial approach, when we come up with 3 different constraints. For a protein to be included in the negative training set of a target Go term, it should satisfy all of these constraints. First one says that Go term annotations of the protein should not contain the target Go term or any of its children. This is pretty straightforward. The second constraint says that if the protein is annotated with the parent of the target Go term, it should also be annotated with a sibling of the target Go term. Here we assume that sibling terms are defining orthogonal functions. They are mutually exclusive. It does not hold in all cases, but often holds. And the last constraint is the protein should have at least 5 asserted annotations by any evidence codes. And by this constraint, we try to avoid those cases where new annotations are added, and we found out that our protein is a false negative, actually. So in the end, we generated our negative and positive training data sets, and the classification methods, binary classification methods takes these into account to classify the protein into negative or positive training data sets. It produces a prediction score between 0 to 1, and prediction scores close to 0 indicates strong negative predictions. Here, in order to test the performance of our method, first we done a benchmark analysis, and the benchmark set was composed of those not annotations in UniProt. We trained our systems without those and used them as our query set. Long story short, by selecting 0.8 as the positive score, prediction score cutoff, and 0.3 as the negative prediction score cutoff, we had a pretty good recall and precision performances. After that, we set out to do a case study, and we take the loss of function into our subject. Prediction of variation impact on disease-related genes is quite important, and we take a CHD8 gene which has been associated with autism spectrum disorder, and there has been some wet lab experiments to show that these two functions, these two gene ontology-based functions, has been partially lost in certain transcripts that has been expressed by the genomes of the patients. So we take the natural variant and also the disease-related variants and run them on our tool, and we find out that our tool able to detect the partial loss in the function. So to conclude, I want to mention our web server. Here, this is the URL. You can upload your protein sequence to get the gene ontology-based functional predictions. And this is also a database of pre-computed predictions for nearly 50 million UniProt tremble proteins. And the tool outputs a prediction score for all of the Go terms. Then you can select your own threshold for positive and negative functional prediction consideration. We also paper came out early in this year in proteins. Please take a look if you are interested. And in the last slide, I want to thank my team members, and thank you for your interest.
Time for one brief question. Otherwise, thank you very much. We look forward to going to your poster. Great. So next one is Pixis map by